Hello, everyone, and thank you for joining the Research Updates from Israel webinar. We're just letting people into the main room now, so we'll get started in just about a minute or so, but thank you all for joining. We'll get started in just a minute. Okay, hello everyone. Thank you for joining our Research Updates from Israel webinar. All participant lines are muted and without video. Please be aware that the taskbar is at the bottom of your Zoom window. This taskbar may collapse when it is not in use. To prevent the controls from auto-hiding, you can use the following keyboard shortcuts to pin your taskbar. For Windows, press the Alt key, and for Mac, press Command and / at the same time. Today's webinar is being recorded and is available with closed captioning. To activate the closed captioning, please click More on the bottom right corner of your Zoom window, select Captions, then Show Captions, and you can also select language there as well. We also have ASL interpretation on the call with us, which will be spotlighted. Throughout the call, you may ask questions through the Q&A and chat features, or by sending an email to info@fightingblindness.org. We will address questions towards the end of the call during the Q&A session. If we do not answer your call or your question live, we will follow up in the days ahead via email. I would now like to turn the call over to the chairman of the Foundation Fighting Blindness Board of Directors, David Brent.
Welcome. I hope you can all hear me. I can tell by the signing that you can, so that's good. I know you're calling in from around the globe. I happen to be in Buenos Aires, so I think all of us we've got several continents handled here. So thank you for taking the time to join us. I'm David Brent. I'm the chairman of the board of the foundation. I'm so happy to be here with you today. Like many of you, I'm so excited to learn about the advances that move us forward for treatments and cures of inherited disease. Forums like this one are hosted today, provide us with the opportunity to hear more about the efforts of the foundation and the partnerships we continue to form around the world. This partnership with many of the researchers in Israel has been more than 15 years in the making. I just was reminded of a visit that Steve Rose and I made back in 2013 probably, when we came up with the idea that we would try to work to have all of the people in Israel affected with retinal disease screened, genetically screened, so that it would be a focal point for research for clinical trials around the world. Foundation is a global leader. It's not just funding critical research for retinal disease, but bringing experts together from around the world to work collaboratively. That is one of our key initiatives for the last 10 years. One initiative is the international campaign to not just raise funds, but to gauge other families. Laura Fiera is on the call today with us from London. And it's our international chair and helped to raise international awareness and much needed funds for the foundation. So thank you for the time. I'm thrilled to introduce to you our Chief Scientific Officer, Claire Gelfman. Claire, over to you, and everyone take care.
Thank you so much, David, for that nice introduction. As David mentioned, my name is Claire Gelfman, and I'm the Chief Scientific Officer here at the Foundation Fighting Blindness. And it is a real pleasure to have all of you with us today, as we are joined by an amazing group of researchers and clinicians from the Israeli Inherited Retinal Disease Consortium, which is a foundation-funded initiative started about 10 years ago. During our forum today, they will cover the research landscape and current progress as they describe their pioneering work in the area of inherited retinal diseases. So in our audience today, we're joined by a select group of VIPs, hand-selected for this exclusive research update. We have found that in these smaller focused webinars, the art of pulling together experts in a virtual room fosters collaboration, as well as identifying and filling in those knowledge gaps, resulting in a greater impact on the field. I'd like to briefly review our agenda for today and what to expect over the next 90 minutes. We're going to hear 5 presentations from our esteemed scientists and clinicians, starting with a presentation on the history of the consortium from Dr. Perlman, followed by learnings from a genetic analysis of IRD patients in Israel from Dr. Ben Youssef. Dr. Sudri will then discuss therapeutic strategies for the treatment of IRDs, followed by Dr. Sharon and Dr. Bannon's presentation on retinitis pigmentosa caused by mutations in the FAM161A gene from patient to bench to treatment. Our final presentation will be delivered by Drs. Rodenstrike and Scher, who will talk about the application of personalized medicine for the treatment of RP, focusing on the retinoid cycle and PRPF31 mutations. At the end of all the presentations, a question and answer period will follow, where you, our attendees, can ask questions of our presenters. We've asked that each presenter stick to their allotted time to allow for 20 minutes at the end for our Q&A. We will then conclude with a few closing remarks from Jason Menzo, the Chief Executive Officer at the Foundation Fighting Blindness. So without further ado, it is my esteemed pleasure to welcome Dr. Edu Perlman, Director of the Consortium, who comes to us from the Technion Israel Institute of Technology, who will speak on the history of the consortium, goals, and modes of action. Dr. Perlman, the floor is yours.
Dr. Perlman, you're muted.
Okay, I was muting it. Good morning to all of you and good evening to us. And it's my pleasure and my honor to start this webinar presentation and to start and speak and give you an explanation about the history of this the Israeli Inherited Retinal Disease Consortium. We started our activity at March 2014 and ended it really in type of financial at September 2020, but we are still going on, and as I'll explain later. Our goal was to, our major goal was to do the mapping of all or the major part of the Israeli inherited retinal degenerative disease in order to be able to support these patients, to improve the situation, to get smaller number of IRDs in Israel, and to research for some therapeutic. So we started. We wanted to do this mapping in order to get prevention and counseling. Basically by the genetic counselors in order to prevent the growth of the number and to do counseling for those who are affected. We wanted to have this once mapping the entire or most of the patient in Israel, our patient can volunteer and can participate in clinical trials when there are clinical trials, because there are not that many patients in the world having the same gene mutation, and that was one of the goals. And the other goal is that our patient will be able to get as fast as possible the approved treatment. So when something like Luxturna come out, we can look at our database and see which patient is ready for such treatment. And then we wanted to have research intended to develop treatments for genes that are unique to Israel. And you'll hear about it today. We have more specific goals, aims. We wanted to determine the prevalence of inheritance patterns in Israel and about our ethnic groups. We wanted to discover new genes and new mutation in known genes. We wanted to know the prevalence of genes for each Israeli ethnic group. And we wanted to learn the phenotype-genotype correlation of the Israeli IRD. There is a big advantage to start such a program in Israel. The population of Israel is about 10 million people, and they are located in a relatively small geographic area. We have many segregated ethnic groups. Jews, we have Ashkenazis, North African, Yemenite, and we also find that Jews from Georgia, they tend to marry within the group, or from Bukhara, they tend to marry between the groups. So there are many ethnic groups to be studied. The Arabs, we consider as Muslim and Christians. We have Druze, Bedouins, and many more. So it's a very rich ethnic genetic background. There are specific founder genes for each ethnic group, so it's good to learn them. And beside all this, on top of all this, the ophthalmologist in Israel is highly advanced, and we have some excellent genetic centers that are spread all over Israel to reach the Israeli population. And here is a map of the groups of the consortium. We have right now, we have 5 clinical electrophysiology of vision groups. Eyal Banin is in Jerusalem, and Hadass Neumann and Diana Tzur and Yigal Rothenstreich and Miri Ehrenberg are 3 groups in the center of Israel, in Tel Aviv area, and Rina Leibo is in Haifa. So they are spread from north to south, to almost south. We have several genetic centers, Dror Sharon in Jerusalem, Hagit Bris-Feldman in Tel Aviv, Eran Pras in Tel Aviv. We have Tamar Ben Yosef from Haifa, and Ohad Birk and Libe Gretstein in Be'er Sheva. And then we have one bioinformatic group, Noam Shomron, who is located in Tel Aviv. So we are all spread very well in Israel. But one of the biggest problems was to find the modes of recruiting patients. Once we started to work, we had many patients with known diagnosis. We wanted to reach them and to bring them for genetic diagnosis. So the ERG-VP centers, the electrophysiology centers, we had many patients in the Eyal Banin lab, in my lab, in Yigal Rothenstreich. We can go in our database and find them and try to convince them to come, because it will be for the benefit. And each one who came in was sent to the genetic center for analysis. And then the ophthalmologists from HMOs, those working in the field, when they find a new suspect for IRD, they send them to the ERG center, who will send them to the genetic center. Then sometimes the ophthalmology in the field, in the HMO, will send directly a patient to the genetic center. Now, depending on the genetic center, the type of patient, they may send them for phenotype analysis, and then we send them back to the genetic center. Lucky for us, we had the Lirot, the group from the Israel that is the counterpart to FFB in Israel, who is supporting research in vision. They organize special meetings of interest for interest groups, meeting for RP patients, for Usher patients, and others. And they were organized all over Israel, from Haifa to Be'er Sheva. And you see one of the groups here sitting and listening. And then there was some self-referral. Patients read about us in the Israeli press, and they call and say, yes, I am IRD, I want to have a genetic analysis. And they came to the genetic center, who sent them to us, to the ERG-VP center, or did not. It depends on the phenotype determination. I want to give you, Israel is, as I said, it's a small country, many people, and a lot of ethnic group, and we wanted to have a rough estimate. And this is a very, very rough estimate. Dror Sharon is working now on a much more precise prevalence data, but I'll give you just a rough one. So until today, we recruited about 4,500 families with IRD, and they all underwent or gave blood for genetic analysis. And then I made one assumption that each isolate family include only one IRD and all the others include at least two. Sometimes I'm wrong, but sometimes there are many more than two. So I think I'm not that far from the truth. And what I got is that if I average, we have 26% isolate and 74% all the rest, which meaning autosomal recessive, autosomal dominant, and sex-linked. And so the analysis came out that the average number of patients in each family is about 1.74. And if I take 1.74 multiplied by 4,500, which is 7830 IRD patients, and this is all IRD, not just RP. And if I look at the Israeli population, which is 9,700,000, I come up with a number which is 1 out of every 1,240 people. This is a very high prevalence. And I think I'm not wrong by much. It can be 1,000 and 1 to 1,500, but that's about the range of precision. And this is a very high number. And this is, and you'll see later where does it come from. I want to give you a little bit explanation. How did we work and how did we learn just from looking at the patients? So at the beginning, when we start, look at the blue. This is when we collected most of the data. We had quite a few autosomal recessive and a lot of isolate. Few dominant and few sex-linked. Once we start gathering data and evaluating the genetics, we realized that most of the isolate were really autosomal recessive. And as you see today, we have a lot of autosomal recessive, which is the red, very few isolate, the same autosomal dominant, and a little bit more sex-linked. So we can just by having mapping, the first mapping, we can get differences in the type of in the, how you say, the inheritance pattern. Another thing, what we did, we took the number of patients from each ethnic group and compared them to the number of patients and to the number of population in Israel. So there's or to the number of IRD. And you see that for Druze, they are about 1.6% from the Israeli population, and they have about 2% of the IRD group, which makes sense. Christian Arab is also similar, 1.9% from the population, 3% a little bit higher in the IRD. Christian Muslim is very high. We have 18% in the population and 31% of IRD, and Jews is 73% of the population and 64% of the IRD. The Muslim Arab is really an interesting group. It turns out that they have a very high rate of consanguineous marriage, and they have a lot of autosomal recessive, and the IRD is much more prevalent in the Muslim group. I just want to finish with this slide, and it's a summary for all what you hear later. We was funded by FFB and the Israeli Ministry of Health for a total of about $1.3 million from March 2014 to September 2020. However, we didn't stop working. We are working still, and all the consortium is still alive. The groups, the centers are working together on the specific aims that I listed before. As a matter of fact, 2 weeks ago on Thursday, the May 11, we gathered for our annual scientific day. demonstrating the innovative research that is carried out in the consortium center. We would like to thank the FFB for the generous financial support. I'm not taking it for the end. I want to say it at the beginning, and specifically to Dr. Amy Laster for the understanding of our needs, being flexible with our request. And I would like to thank Lirot organization for helping us. They help us a lot to secure the financial support from the Israeli government and for organizing 6 meetings of IRD. So this is my presentation, and the next one will be Professor Tamar Ben Yosef from the Technion. And Tamar, the floor is yours.
Thank you, Ido. Just a second here. Yes. Okay, thank you very much, everyone. So I will talk about genetic analysis of patients with inherited retinal diseases in the Israeli population. What have we learned? And on the map to the right of my slide, you can see the distribution of IRD patients that we ascertained for this study. And you can see that they're really distributed throughout the country. As for the distribution of ethnic groups, you can see that the 2 major ethnic groups in our cohorts, the blue group and the red group, are Arab Muslims and Ashkenazi Jews. Now, Ashkenazi Jews, it's not surprising because Ashkenazi Jews are the major Jewish ethnic group in Israel. However, as for the Arab Muslim group, as Ido already mentioned, they account only for about 19% of the Israeli population. However, they account for 25% of the patients in our cohort, meaning that they are overrepresented in our cohort. And the main reason for that is that among the Arab Muslim community, it is very common to marry with relatives, consanguineous marriages, meaning that people marry with people that they are related to. And consanguineous marriages increase the rate of autosomal recessive diseases, diseases in which you need both copies of a gene to be mutated in order to represent to show the disease. And many autosomal recessive conditions are common among Arab Muslims due to this cultural fact. And among them are IRDs, and this is why they are overrepresented in our cohort. What about phenotypes, types of diseases? So the major disease we see in our cohort is retinitis pigmentosa, RP, 43% of the patients, and this is not surprising. We see it also in other cohorts throughout the world. This is followed by Cone and Cone-Rod dystrophy, Stargardt disease, which is a type of macular degeneration, and Usher syndrome, which is a combination of RP and hearing loss. What about the inheritance pattern? So by far, as you can see here in blue in this pie, autosomal recessive inheritance accounts for 61% of our patients, and this is due to 2 main reasons. As I mentioned already, the high degree of consanguineous marriages in the Muslim Arab population, and among Jews, the existence of common founder mutations in specific ethnic groups. So Jews in the diaspora, before they arrived to Israel, they used to live as close communities which didn't mix with the surrounding communities. And as a result, each Jewish ethnic groups, wherever they live, developed their own set of specific mutations, and which became quite common in this population. We call those founder mutations, which means that nowadays, in Israel, Jews almost don't marry with relatives. It's very uncommon among Jews. However, if a Jew in Israel marries another Jew from the same ethnicity, for example, an Ashkenazi Jew marries an Ashkenazi Jew, or a Moroccan Jew marries a Moroccan Jew, there is not a small chance that they will both be carriers of the same mutation, even if they're not directly related. And this brings to this high degree of autosomal recessive IRDs. As for the genes underlying IRDs in our cohort, the 2 major genes are ABCA4 and ASH2A, and this is very similar to finding throughout the world in North America, in Europe, and in other places. However, when we look at genes number 3 and 4, here we can already see a difference. At number 3, we find the FAM161A gene, which accounts for 6% of our cohort. while in other cohorts throughout the world, it accounts for less than 1%. And the reason for this gene being so highly represented in our cohort is that when we look at the patients with mutations in this gene, we see that almost 90% of them are North African Jewish descent. And in this ethnic group, there are two common founder mutations in this gene, which make it so common in our population. And you will hear more about it from Eyal Banin and Dror Sharon. As for the fourth position, we find the CNGA3 gene, which accounts for 5% of our patients. And again, in other cohorts throughout the world, we find it in less than 1%. And the reason here is a common founder mutation, which is very ancient, and it is shared surprisingly among two different ethnic groups, Arab Muslims and Oriental Jews, and they have the same mutation, implicating that maybe they actually have a common origin, something to think about. We use this very unique population that we have in order to conduct very specific and targeted studies on different ethnicities. And this is an example for a study that we conducted on non-syndromic retinitis pigmentosa among Ashkenazi Jews. And we can see in the chart that the two major genes that account for RP in Ashkenazi Jews are MAC and DHDDS. So we can use this information, of course, to provide the Ashkenazi Jews in Israel and in other places in the world with specific genetic counseling and advice. However, Ashkenazi Jews reside in Israel as well as in North America and in Europe. But in Israel, we also have very unique ethnic groups that are found only in Israel or almost only in Israel, and we need to study them in order to provide them with the information. And one example is Jews from the Caucasus. The Caucasus, as you can see on this map, is a geographic region between the Black Sea and the Caspian Sea. And many Jews used to reside there until in the 1990s, after the collapse of the Soviet Union. Most of the Jews that used to live there immigrated to Israel. And to date, most of them live in Israel. And in this study, we found that Jews from this ethnicity that have retinitis pigmentosa, most of them have the same mutation in the PDE6B gene. And this enables us now to provide patients from this ethnicity very specific genetic testing. Another example are Jews from Ethiopia, also known as Beta Israel. They used to live in Ethiopia in many different small villages, and they started immigrating from Ethiopia to Israel around the 1980s. And nowadays, there are about 160,000 Ethiopian Jews living in Israel, which is about 1.5% of the Israeli population. However, even to date, most of Ethiopian Jews marry within their community, but very little is known about the genetic basis for various genetic conditions, including IRD, in this population. So we performed a specific study on the genetic causes for inherited retinal diseases among Israeli Jews of Ethiopian ancestry. And we found that the most common phenotypes are RP and Stargardt disease, which is similar to what is found in other populations in the world. Also, the most common gene is ABCA4, which again is also found in other places in the world. But the difference is the specific mutations that we found. Most of them are mutations that are very rare or do not exist at all in other populations and are unique to the Ethiopian Jewish community. And due to this study, now we can provide Ethiopian Jews residing in Israel very specific genetic testing and counseling, and hopefully therapy as well. I want to summarize by saying that over the years, members of the Israeli Inherited Retinal Disease Consortium were involved in identification of over 20 novel genes underlying IRD, which is 7% of the IRD genes known today. And this is impressive because the Israeli population is only about 0.1% of the world population. So we are only 0.1% of the world population, but we contributed 7% of the known IRD genes. And this is due to two reasons. One is the unique structure of our population, which enables us to find very unique findings that are harder to find in other places. And second, because we have excellent medicine and excellent research, and we know exactly what we're doing, so we can utilize this precious resource that we have and use it to the benefit of our population as well as other populations throughout the world. Thank you very much. And our next speaker will be Dr. Shiri Sudri from Rambam Medical Center in Haifa.
Thank you very much. Hi, everyone. Okay, so I'll share my screen. Okay. All right. So thank you, Tamar, and thank you, everyone, for joining us. And I think when you think about the situation when a diagnosis of inherited retinal degeneration is established, and by the way, from now on, I will not say inherited retinal degeneration, I will just say IRD for short. So once a diagnosis of IRD is established, the next question that comes up is what can be done? And it is my pleasure to share with you this evening, some aspects of the developing therapeutic options and directions. So as you all know, IRDs are actually a group of diseases which can be very different from one another, but all of them are characterized by the progressive loss of the photoreceptors and the cells in the retina, including the retinal pigment epithelial cells, or RPE, which basically lead to vision loss. Now, until now, IRDs have been considered incurable, but we're very fortunate to be witnessing the beginning of a historic shift in this regard, because with the recent rise in research efforts in relevant fields all over the world, several treatment options for IRDs are now becoming available on the horizon. Now, largely speaking, the leading therapeutic directions for IRDs include mainly gene and cell therapy. And the eye is an organ which is very suitable for such therapy. First, it is small and can be easily accessed. It needs a relatively small volume of the injected material is required. Typically, an immune response to the genetic or cellular materials which are being injected is relatively limited, and so is the systemic spread from the eye, which is an important safety factor. And finally, advanced imaging systems of the retina, which are very commonly available, enable us to monitor the response to treatment in a non-invasive manner. And indeed, over recent years, there has been a significant surge in research of new genetic therapies for IRDs, with an impressive rise in the number of participating subjects and centers all over the world. Now, when we speak of genetic therapy, it's interesting to note that there are several possible approaches. Classic gene therapy, which is also called gene replacement or gene augmentation, basically means insertion of a functional copy of the mutated gene to the target cells. And in contrast, genome editing using novel approach called CRISPR is a different approach which enables to edit the human genome within the affected cells, in this case in the retina. Now, in either case, gene therapy requires a vector for the transfer of the genetic material into the target cells. And in the case of retina, adeno-associated virus, or AAV for short, is considered the safest and most effective viral vector platform, at least to date. And there are certainly clinical trials for several genes which are involved in different IRDs, which are presently underway. And on this slide on the right, I've listed a few of the conditions, including Leber congenital amaurosis, choroideremia, retinitis pigmentosa, Usher syndrome, and achromatopsia with the involved genes, which are listed as well. And of course, we cannot go without mentioning RPE65, which is a gene which encodes a protein which plays a key role in the visual cycle and is therefore very important for the entire visual process. Now, defective RPE65 gene leads to an early onset retinitis pigmentosa and Leber congenital amaurosis. And as you all know, in December 2017, the FDA approved the first gene therapy, which is called Luxturna by Spark Therapeutics, to treat subjects with RPE65 related or associated retinal dystrophy in the US. And the situation in Israel is following. The drug is now also approved in our country. And so far, 11 patients were treated, mostly at the Hadassah Eye Hospital in Jerusalem. And I think at least some of these subjects were treated directly thanks to the efforts of the Israeli consortium. And for us, it was very exciting and almost thrilling to learn about this privilege to treat patients. Another unique approach for genetic therapy, which was developed in Israel, actually was developed by Professor Dror Sharon and Professor Eyal Banin, whom you'll meet in a short while, together with researchers from Bar-Ilan University, employs RNA editing. Now, this method uses enzymes which are self-occurring to edit mutated RNA, causing a variety of IRD phenotypes. Now, unlike DNA editing, including CRISPR method, which I've just mentioned, RNA editing can be fine-tuned and is less risky. And it is my pleasure to share that the group showed initial success in vitro studies, and therefore in vivo studies in the mouse photoreceptors are presently underway. So hopefully there is a lot of exciting news that are coming up ahead. A different approach for the treatment of IRD is cell replacement therapy, which aims at replacing damaged host cells with transplanted donor cells. Now, for IRDs, this direction has mainly focused up until now on transplantation of the RPE cells, because these cells, as I said, support the photoreceptors and are vital for the retinal function. And in fact, the RPE cells are important for the entire visual process. Now, quite a few different mutations which affect the functions of the RPE can cause IRDs. And one advantage of cell therapy is that this is a mutation-independent approach, which can therefore work even when the cells are completely lost, because the lost cells are simply replaced by the transplanted ones. There are several options for the types of cells which can be used as the source for the transplantation. And the main use, at least in the present time, is of induced pluripotent stem cells, or iPS cells for short, which are becoming a very attractive strategy for IRDs. Now, very briefly, iPS are cells which are generated from mature body cells like the skin fibroblasts or peripheral blood cells, which are collected from the subject and are reprogrammed in the lab to become stem cells. The derived stem cells can be then differentiated again in the lab into various cells which are used for the transplantation, including, for example, the RPE. Now, this approach faces several challenges in the present time, but has a promising potential for IRDs. Now, the transplantation itself can be done either by injection of the solution under the retina, which basically requires a surgical approach, which is done in the operating room, or via intravitreal injection, which theoretically can be done in the office. Each option carries different advantages and disadvantages. And again, it's exciting to share that there are several ongoing phase 1 or 2 clinical trials, which are looking at the efficacy of stem cell therapies for IRDs. And in this regard, it's my pleasure to mention the new Israeli Consortium for Innovative Stem Cell Research for Clinical Use, which was recently established, and which aims to bring together leading scientists and pioneer physicians with a goal to accelerate the development of stem cells for transplantation in various human diseases. And of course, the RPE and retina are included and listed as a top priority. Now, in the future, it seems that a combined genetic cellular approach will become important for the treatment of IRDs. Because if we think about it, if we think about iPS, for example, it's important to note that because these cells are derived from the patient, the cells carry the same genetic risk factors that the patient suffers from, and therefore they need further genetic manipulation to correct the mutation within them and be healthier. And gene editing technologies such as CRISPR and their combination with the development of the iPS cells has the potential to repair patient-specific mutations and opens a new era of personalized precision medicine for our patients. So to summarize, it seems that both the genetic and the cellular approaches hold a very great potential to become successful treatment strategies for IRD patients in the following decades. Now, some of what I say may sound imaginary, but is already happening right now, much thanks to the deep commitment and combined efforts of everyone involved, including you. So indeed, the future is bright. And with this, I thank you for your attention. And I invite my dear friends and colleagues, Professor Sharon and Professor Banin, to give their talk.
Thanks. Hello, everyone. So we're going to go to a double talk now. I will start and Professor Eyal Banin will continue. And we will discuss one gene, FAM161A, which is the major gene for retinitis pigmentosa in the Israeli population. And we will go from patient to bench and hopefully for treatment. So just to, second. Okay. Just to give an overview. So I will talk about a project that started 15 years ago with gene identification. I will discuss the prevalence of FAM161A in the Israeli population. I will present some data which are worldwide disease prevalence that was published in the Proceeding of the National Academy of Sciences about 3 years ago. And then Professor Banin will continue about clinical features of FAM161A, a knockout mouse model that we created, and very exciting results of gene augmentation therapy for this disease. So here at Hadassah, we started recruiting patients about 20 years ago, and we are recruiting about 100 new families every year in a pretty steady pace. We recruited so far almost 2,300 families and over 4,000 patients with IRDs. And using different techniques, we identified the genetic cause of disease in about 65 to 70% of cases. And our goals are to identify the major phenotypes and the most common genes and founder mutations in our populations, and to seek for novel genes mutations and novel mechanisms for IRD. We are notifying the patients once we identify the gene, and we refer them to genetic counseling for disease prevention. And we're using the findings to explore for and apply for novel treatments, as Professor Banin will describe in his part. Over the years, we were involved, or we identified over 20 novel genes. FAM161A was among the first ones that we identified. Some other major genes we identified are DHDDS and MAC, which are relatively common in the Ashkenazi Jewish population. As you can see, we in most of the genes have been identified as part of collaboration with members of the consortium, and especially Professor Tamar Ben-Yosef. So if I go about 15 years ago, when genetics was not as advanced technological speaking as it is today, we tailored the genetic technique we're using to the population we're studying. And as Professor Ben-Yosef mentioned, many of our communities have relatively high level of consanguinity. And once this is happening, patients with RP, many of them will inherit the same mutation from both parents. This will lead to homozygosity, and there is a technique that we used that is based on this phenomenon, which is called homozygosity mapping. So when we used this technique about 15 years ago, we realized that there is a specific region in the human genome in which we suspect that there is a novel RP gene in 3 different populations. One big population was the Moroccan Jewish families. Another one is Jews from Syria, and a Palestinian family. So in this 3 group of families, we identified the same genomic region. However, among the 25 genes in that region, there was no information about any gene that is highly expressed in the retina, and we had to search them one by one. This is how it was done at that time, until we identified the 3 mutations in the FAM161A gene. At the same time, a dear friend from Switzerland, Carlo Rivolta, identified mutations in the same gene in another group of patients from Europe and India, and we published the results together. When we identified FAM161A, there was no knowledge about its expression pattern or function. So we started to make some experiments and reveal some data about the gene. So the first set of experiments was about the expression pattern, and we realized that the gene has dual expression patterns in the mouse retina, part of it in the embryonic retina, and later on its expression is pretty restricted to the photoreceptors. We also identified that the gene can produce 2 different protein isoforms. And this is very important for therapy because we need to know which isoform we should use for gene augmentation therapy, and Professor Banin will touch this point later. We also did some expression analysis of different tissues, and we realized that FAM161A can be expressed in other tissues, but the highest expression level is in the retina. What does it do in the retina? We still don't know for details, but we do know, and this is a work again together with Carlo Rivolta, we identified that it is expressed in a specific part of the photoreceptor cell. This part is called the cilia, and FAM161A is now a member of a large number of photoreceptor proteins that cause ciliopathies. So retinitis pigmentosa due to FAM161A is one of the ciliopathies. As mentioned before, FAM161A is the major gene for retinitis pigmentosa in our population, mainly due to 2 different mutations in the Jewish population. On the left side, you can see a few families with FAM161A mutations, mainly due to consanguinity. And on the right, you can see that most of the patients are from North Africa and Jewish origin. And the 2 mutations that were found in the Jewish population were never reported in patients who are not of Jewish origin. So these are unique for this population. Next step, we wanted to evaluate what is the prevalence of IRDs worldwide and the prevalence of specific genes, including FAM161A. So we initiated a very large study in which we studied about 10,000 mutations that can cause IRDs. And I'm not going to go into too much details, but just to show that we were able to calculate the expected number of patients with IRD in every population that we studied worldwide. For example, we predict that there are about 9,000 affected individuals with IRD in Africa alone, and we have this calculation for each population. And we were able to do that for specific genes. So if I will go to my last slide, you can see the prevalence of 2 genes, RPE65 at the bottom, and this is the gene for which Luxturna, the gene therapy, has been developed. And you can see that there are about 15,000 affected individuals that are expected to be to have LCA or RP due to RPE65. By the way, most of the affected individuals are expected to locate in Africa, and therefore many of them are unlikely to benefit from the gene therapy. However, with FAM161A, the picture is a bit different. There are only 3,600 individuals with who are biallelic for FAM161A, about 1/3 of them from Europe. And this is a challenge when one wants to develop gene therapy because the number of affected individuals is not as high as other genes such as RPE65. I would like now to introduce a dear colleague and friend of mine, Professor Eyal Benin, to continue telling you about the story of FAM161A. Thank you.
Hello to everyone. Thank you for joining, and I'm really very, very glad to be here and to be able to continue and share with you our journey that started with the identification of FAM161A as an RP-causing gene by Dror in 2010. And once we identified this is the cause for RP in our population, and not only the cause, the one cause, but actually the main cause for recessive RP in our population, we started collecting these patients, again, through the efforts of the consortium. And thanks to these efforts, we have now more than 140 patients diagnosed, and the study I'm going to describe now was done a few years ago in order to try to establish the clinical phenotype of the patient. So in many ways, it's similar to other forms of RP, but on the other hand, it has some specific characteristics. And the reason we wanted to clarify the phenotype was to see if there is a window for treatment, possible window for treatment in these patients. You know that if the disease is too early and too aggressive, one might have difficulties in providing treatment, but if there's a window in which some vision is preserved, that allows the opportunity to try to develop treatment. So the age of onset of FAM161ARP is between the first and third decades. The initial symptom in most patients, above 70%, is night blindness. This is followed by visual loss, and the decrease in visual acuity actually occurs rather late, which points to the fact that we might have a window for opportunity for treatment, and most of them are also myopic. And the next slide shows you plotting of the visual acuity of different patients with FAM161ARP versus age. And to cut a long story short, in the cohort of over 100 patients, many patients still maintain visual acuity of over 20/50 up to the 5th decade of life, again, offering perhaps window for treatment. This is some fundus imaging of FAM161A patients, and in many ways it's similar to other forms of RP, but notably bone speckle-like changes, which are one of the hallmarks of the disease, are relatively mild in most patients up to 40-50 years of age. And in many cases, this also leads to some delay in the diagnosis, because when one looks at the fundus at early ages, you don't really see this characteristic sign. In late stages, again, the pigmentary changes do appear. The OCT findings in the patients support, again, a possible window for treatment, because while there is increased thinning of the nuclear layer, the photoreceptor layer, over time, there are, in many, many cases, relative preservation in the foveal area where we have our high-resolution viewing up to the 5th decade, correlating with what we saw in the visual acuity data. And another thing that sort of protects these patients from changes in their central vision is the fact that cystoid macular edema, which is cystoid changes and edema within the center of vision, is quite rare in FAM161 patients. Visual fields do constrict very early, even during the 2nd decade of life. However, this small island is maintained for many, many years. The results of the clinical phenotyping showed us that it is, first of all, a leading cause in our population, and that in many cases, a small central island of photoreceptors around the fovea that is maintaining good vision is preserved up to the 5th decade, and there seems to be justification and opportunity to try to develop treatment. The next slide sort of gives you a rough roadmap of what one might think to do as a roadmap towards treatment based on gene augmentation, as Shiri mentioned earlier. In other words, delivering a normal copy of the gene in order to provide the cells with the gene that they're lacking because of the mutations. So once you have your disease-causing gene identified, the next thing you might want to do is to find or develop an animal model in which you might test your therapy. Then you would go on to develop a viral vector to deliver the gene to the retina. You would need to deliver it in the animals and examine whether this is efficient and safe. And if this is so, one would then move to the translational research phase, which means making clinical grade vector, validating its efficacy and safety again in the animal model, and hopefully reaching a phase 1 trial. So a very talented PhD student in Dror's lab actually established a knockout mouse model of our FAM161ARP. In other words, she knocked out the FAM gene, so it was not expressed at all. And indeed, these mice showed no expression of the FAM161A protein in the cilia of the photoreceptors. And when we started to characterize the course of disease in these mice, it seems that they actually manifest disease that is quite similar to that we see in patients. Here you see the tests we use to assess visual acuity in mice based on the optokinetic response. These white and black bars move, and we can see whether the mouse is following it through the tracking of his head. And you can see here that between the age of birth and the age of 8 months, these mice slowly lose their visual acuity, and by 8 months they cannot follow the grid anymore, even when the bars are very wide. Similarly, when we tested the electrophysiological function of the retina, we see that over time, the response to stimulation of light, the electrical response of the retina, drops, and by 6 months, the responses are barely detectable, and they become non-detectable at 8 months. This is showing it over time by the different colors. On OCT imaging, one can see that over time, the layer of photoreceptor nuclei, the ONL as it's called, slowly thins, and This is further supported by histological sections showing that the ONL gradually thins until by 8 months we see only sparse photoreceptors, and we also quantify this. So we now have a very good model that seems to emulate the human disease. Now we need a viral vector to try to deliver the correct or the intact FAM161A gene to the retina of these mice to see if we can indeed attenuate the course of disease. And here, thanks to a very fruitful collaboration with a group in Switzerland, Dr. Ivan Arstenievich and Dr. Corinne Kostik, who are experts in the production of viral vectors, they produced viral vectors that contain the FAM161A gene. And you know that gene augmentation therapy today in the clinical arena is based on using viruses that do not replicate and do not cause disease, but they are sort of a Trojan horse in a positive sense of the word, in that they deliver the genes to the photoreceptors and the cells of the retina. And what we do is we inject these viral vectors under the retina. You see here a picture in the mice. It's challenging, but it can be done. And then we follow to see what are the effects of this treatment on the course of disease in the mice. So this is imaging similar to what we do in patients. This is autofluorescent imaging in mice. And what we did is that in some mice, we treated only half of the retina. And in some mice, we did a double injection and treated both halves of the retina. So on top, you see changes in autofluorescence imaging over time in an untreated retina. And you see that over time, we see these hypo and hyperfluorescent spots appear that are part of the degenerative process. When we do a single nasal injection and treat only the nasal half of the retina, as marked by the green arrows, you can see that these changes do not really show up even up to 8 months, while the untreated side shows these changes, giving hope that the treatment has indeed slowed down the disease in the treated area. When we do a double injection and treat both sides of the retina, these changes in the autofluorescence imaging are attenuated significantly. And similarly, on OCT, we can see better preservation of the photoreceptor layer in the treated side in single injected retinas, and in double injected retinas, as compared to the fellow eye of this animal, one can see that the ONL is indeed better preserved. So looking at actual visual function, again, the optokinetic response, the loss of vision over time, you can see that in the treated knockout mice, which is the green bar at 8 months, they're still maintaining rather high level of visual acuity, while untreated mice have lost it completely. And the same holds for the ERG responses that still do deteriorate, but much more slowly, and visual function is maintained well over 8 months. On histology is perhaps the most convincing evidence, because here we can see the photoreceptor layer, the ONL, and an enlargement is seen in the middle, including a graph that measures the thickness of this layer. And we see that in a single injected half-treated retina eye, the half that is treated has preservation of the photoreceptor layer, while the contralateral side, which is not treated, we have lost most of our photoreceptors. And when we do a double injection, which is shown in the retina in the bottom, there's good preservation of the photoreceptor layer. And on top, sort of upside down, is the fellow eye retina, where most of the photoreceptors are lost. And the graph on the left shows the thickness in green in the treated eye versus that in the untreated eye. Immunohistochemistry shows that in the treated region, we not only get rescue, but we actually get expression of the FAM161A gene, which is actually absent in these mice because of the knockout of the gene. And it is expressed in the correct area within the photoreceptors. And together with it, we see the expression of other photoreceptor-specific proteins like CRX recoverin and the opsins, showing that these photoreceptors can indeed be functional, as we saw in the other testing. So to conclude, gene augmentation therapy using the long isoform of FAM161A leads to significant slowing down of the degenerative process and preserves retinal function in the FAM161A knockout mouse. The rescue effect is long-lasting, although it doesn't completely prevent the slow loss of photoreceptors over time. And we hope that the results obtained will serve as a step towards future clinical application of gene augmentation therapy in FAM161A. And if we're talking about the roadmap that we spoke before, we're here at the gate of trying to mount the translational research challenge, which is to make a clinical grade vector, validate efficacy and safety, and move on, hopefully, to a phase 1 clinical trial in humans to this gene that is highly prevalent in our population. And thanks to the consortium efforts, we have now mapped a large number of patients with this disease. This is just to acknowledge the many who helped in this sort of 12-year journey. And it's now really my pleasure to introduce the next speakers, which is Dr. Igal Rothenstreich and Ifat Shel, who will present their effort to characterize PRF31 patients and new treatments in this regard. Thank you very much.
Hi, good evening. My name is Professor Rothenstrach. I'm the head of the electrophysiology clinic and retinal research laboratory, and I'm a clinician researcher.
And my name is Dr. Ifat Shel. I'm head of the Restorative Retinal Research Laboratory at Sheba, and we're both affiliated also with the Sackler Faculty of Medicine at Tel Aviv University.
Vitamin A is a key role in vision, and the cis form, as you see here, is crucial for vision. When the light strikes the photoreceptor, it changes the cis form into trans. And then we need to recycle it via the retinoid cycle, which includes many enzymes, including the RP65, the LRAT, and many more. Mutation in these enzymes can cause retinitis pigmentosa. To overcome the lack of cis form in this patient, we can supply the cis form from the outside. However, there are not many sources, or not even at all sources, of cis form in nature. So I'm going to start a different story, which started 3,500 years ago during the exodus of the people from the Israeli people, the Jewish people that ran away from Pharaoh, and they actually landed in the Bardawil Sea. Later on, during the Yom Kippur War in 1973, a student from Weizmann Institute landed again in the Bardawil Sea and found a seaweed which contains more than 50% of 9-cis beta-carotene. Then we use this seaweed and produce capsules and treat patients with RP, with retinitis pigmentosa. Those capsules contain more than 50% of the cis form. It went to the liver, to the blood vessels, and then reached the eye and converted from the prodrug, from the 9-cis beta-carotene to cis form. We showed improvement in the retinal function compared to placebo in some of the patients, 30% of the patients, 1/3. And moreover, the visual field in some of them improved by 300%, as you see in this slide. However, due to global warming, the seaweed started to produce only 20% of the cis form, and this was not enough. Therefore, we developed a pure synthetic formulation of 9-cis beta-carotene with the collaboration with the Bolvatnik Center. And we succeeded to show that the synthetic form improved the survival of the photoreceptors. But moreover, it also improved the immune reaction during the disease. And this is one of the crucial issues that nowadays all investors are dealing by lowering and regulating the destruction by the immune system in retinitis pigmentosa. Now we are moving toward upscaling the manufacture so we can give it into preclinical studies in animal models and in other issues. Now, just as Dror and Eyal said that about the ciliopathy, we found that the 9-cis beta carotene also is probably needed because the ciliopathies are also harming the enzymes that are trafficking by the cilia. So this treatment can also help in ciliopathies.
Okay, and in the last couple of minutes we have left, I'll tell you a little bit about our approach for disease modeling and developing treatment for RP using mini retina that we grow in a dish that is derived from a patient's own cells. And the first gene we decided to focus in this project is PRPF31. And really thanks to the consortium in Israel, we were able to identify 11 families in Israel that have mutations in PRPF31. And one of the problems in developing treatment for this gene is that animal models do not develop RP. So if you mutate this gene in mice or even larger animals, they don't have retinal degeneration. So to overcome this challenge, we decided to develop a pipeline where we take a tiny skin biopsy from patients. We take cells from this skin biopsy. We turn them back to stem cells, like in the embryo, and then we differentiate it. We take these stem cells, we differentiate them, we mature them to generate a mature or early retinas derived from these patients. So we develop in the lab both photoreceptors and the supporting retinal layer, which is called RPE. And what can we do with these mini retinas that we grow in the lab? First of all, we can understand better the disease because we have our specific patient cells growing in the lab, and we can start seeing what's wrong with the cell, what is the genetic defect doing to the cell, and potentially also develop treatment that are designed specifically for these patients. And in this slide, I'm showing some example of our work. So on the left, you have an example of retinal tissue that we grew from 2 patients that have mutations in PRPF31. Because of the mutation, there is very low expression of the PRPF31 protein in the patient's retina. And I hope you can appreciate that when we look at the cells from the patients, indeed, there is very low staining for this protein. And you can see very low red color in the cells, in the retinal cells derived from these 2 patients. And when we look at the tissue that was generated, we see that it is very sick. The RPE, the retinal epithelium tissue, did not develop well from these patients. Unlike the retina tissue that was developed from a healthy control, which is intact and the cells look very nice and hexagonal shape, the patient's tissue looks sick. We have white holes between the cells, areas that the cells did not mature well. So the next step is how can we help these patients? How can we increase the expression of this mutated of the PRPF31? How can we increase the expression of this protein in order to rescue the disease phenotype? And our strategy for increasing, so there are several strategies that you've heard. One approach is to do a gene therapy, to make a genetic construct and add the gene from outside using a virus or other ways. But our approach is a little different, and it's based on a strategy that is called drug repurposing. And the idea is that we can maybe find a drug that is already in use in the clinic for other diseases, and it may enable us to treat RP. And to test this theory, we developed in collaboration with the Blavatnik Center for Drug Discovery at Tel Aviv University, a special cell line that enabled us to screen thousands of molecules, thousands of drugs that are already in the clinic, nearly 7,000 drugs. And using this screen, we identified 2 drugs, 2 promising drugs that seem to overcome some of the mutations of PRPF31. And this is an example of 1 patient and 1 drug that was found in this screen. So we're looking at a retinal tissue that was derived from the patient, and because of the mutation in the PRPF31 gene, there is hardly any protein expressed, very, very dim red staining compared to the control retina. However, when we add one of the drugs that we identified in the screen, we see nice elevation, nice expression of the protein. So we could rescue the protein low levels by adding the drug. And importantly, we can rescue the structure of the retinal tissue that we grow in this dish. We don't see these holes, these unmature cells. The tissue looks intact and nice and healthy. So our next step in this strategy is first of all to understand the mode of action, how this drug is affecting the cells, how it's helping them to recover from the problem of the mutation. And of course, the next step will be a formulation, a drug that we will be able to give to patients. Because again, the whole idea is that these drugs are already approved for treatment of other human diseases. So we hope we'll be able to treat these patients earlier than if we would have started with a new drug that was not tested before. So of course, all this work would not have been done without our excellent team and the generous support of different foundations that supported this study. But I think very important is the consortium, because thanks to the consortium, we were able to actually identify the patients that have the mutation in this gene. And also thanks to the help of all our collaborators in this consortium, we were able to recruit, enroll patients to these studies. And of course, this also led to open the door to collaboration with other centers, including the NEI, University of Utah, with whom we collaborate on the PRPF31 project and the beta-carotene. So thank you very much.
Thank you.
Hi, this is Amy Laster, Senior Vice President of Science Strategy and Awards with the Foundation Fighting Blindness. I want to first just start by saying thank you to all of our clinicians and researchers that did a wonderful job sharing about all the really great things that are happening in Israel in terms of research for IRD. So thank you again. This time now is the beginning of our Q&A phase. And I just want to remind our attendees that if you have questions, please put them in the Q&A feature that you'll find at the bottom of your Zoom for most people, or within the chat. Or if you want to even email the foundation, you can email the foundation directly as well. But this time is for you to ask your questions. So let's see. One question that we do have, any future plan work involving AI to search through existing drugs or building novel proteins or compounds for treatment?
Yes, so the answer is yes, certainly. Once we have this pipeline of this reporter cell and patient cells, we're doing several things. One of them was to screen the current approved drugs, which is about 7,000 drugs. Apparently, there aren't so many drugs approved for human use in the world. And the second approach is really to what we call medicinal chemistry. So once we find a drug, maybe it's not that active, but we can change it. And first we do it using mathematical models, computerized models, and then we can generate new and what we call new drug entities in order to find maybe a better cure. And again, and the second approach that was indeed asked is the AI approach. And for that, we're collaborating with a researcher in Bar-Ilan University. And it's not on the PRPF31, but on 2 other genes. And we're really using AI tools to model the proteins that are affected, and using an AI find how the mutation affected the protein in each patient, and using an AI approach, screen for 1,000 or 100 of thousands of chemical entities that are out there, not necessarily approved as drug, and see which one of these small molecules can, on the computer first, correct the mutation, and then we can use the cell approach to test it and the patient cells in order to test it in specific patients.
Thank you, Dr. Scher. See, are there any other questions? And perhaps for our research team in Israel, as you know, you're identifying a lot of these most common or frequently occurring IRDs. how do you prioritize those that you will pursue therapeutic strategies for?
Can you repeat the question?
So for the IRD genes that have been noted as having high prevalence, there's more than a few, and we heard about 3 today. How do you prioritize those that you focus on to develop therapeutic strategy?
I can say for myself that it's really a matter of how common they are, because if you find a certain mutation in only a few patients, it can be a very interesting mutation. And of course, for this family or this patient, it would mean the world if we develop a therapy. But being realistic, this costs money, and it is very hard to get funding to develop therapy for a gene that is very rare. Therefore, genes like FAM161A or CNGA3, where you have a bulk of patients in Israel, and you know that you would have a relatively large group of patients that can benefit from that, this is the top priority, obviously. It doesn't have to be necessarily in Israel. If I know that there is a mutation in Ashkenazi Jews that is common in Israel, but it is also common in the US among Ashkenazi Jews and so on, then I will definitely prioritize it, and I'm doing it these days, indeed.
In general, it's very hard to prioritize patients who are very rare. Even the ones who are more frequent are still rare. So I don't have a real priority. I mean, I found a patient, we found something by chance, and we are continue with what we have, what we find. It's not. I don't find myself prioritizing by the frequency of the gene.
And maybe I just add one more thing. One of the reasons where we are looking more like at drugs and not gene therapy is really to think about more like a gene agnostic approach. So for example, the beta carotene not necessarily will work for all RP patients, but it at least potentially would be applicable for patients with mutations in several genes of the retinoid cycle, several ciliopathies. Specifically the PRPF31, some of the drugs that we found may be applicable for other genes as well.
Yeah, this is exactly what I wanted to emphasize as well. So on the one hand, we're trying to look at genes that are, prevalent in our population, and we feel sort of obliged our patients to try to find treatment for these genes. But on the other hand, for instance, the ADAR project, the RNA editing project that Shiri mentioned, is something that we're trying to develop in order to address a larger number of patients, because this might be applicable to a large number of certain types of mutations in different genes worldwide. So there's kind of a combination of what seems to be amenable for some kind of treatment. Prevalence does come into play, because obviously you would want to try to improve the vision in a larger number of patients. And then the third thing is having a window of opportunity. Some of the diseases manifest very early and severely, so the development of treatments should be focused on maybe other, you need to find novel ways to try to do that, and others might be more amenable and have a longer window for treatment, might be easier to treat.
Thank you for that. Another question is just really around Usher syndrome. We know that there's different subtypes of Usher syndrome, but specifically even for Usher type 1C, are there any research or clinical studies that specifically is being done with Usher subtype 1C?
Actually, there is even a unique ASH1C mutation that was identified by Dror Sharon several years ago in the Yemenite Jewish population in an alternatively spliced exon. So we do have this unique mutation, but I'm not aware of a specific study targeting ASH1C at this point.
Actually, there is a study by Uwe Wolfram.
Uwe
from
Wolfram,
Germany.
right?
He developed a pig model for H1C, and he is going to apply gene therapy.
Thank you. And Dr. Sherrill, don't go away. I have another question for you. Can you talk a little bit about creating the clinical grade vector for FAM161A?
Yeah, this is actually where we stand right now. So together with Professor Ivan Arsenevich from Switzerland, we are about to ask for funding for this project because this is really costly. And this is actually our next step. And we're hoping to develop this vector and to test it for toxicity in non-human primates. And if everything goes well, then we will be ready to jump and start treating patients. So next step is indeed to have a vector that is ready to be used.
Thank you. So we still have time for a couple more questions. So certainly if you have them, please put them in the Q&A or the chat feature. And while we're waiting for individuals to kind of ruminate on what they want to ask, I do just kind of want to open back up the floor to our clinicians and researchers. If there's anything following this presentation that you do want to share with the audience, feel free to do so. I'll give you a chance to just have open dialogue.
I can mention that there are a few challenges in the IRD field, as I'm sure you're very well aware. And among them, I hope we were able to convince you how important it is for each IRD affected individual to be genetically diagnosed, not only for genetic counseling in his or her family, but also to know if they can benefit from a certain genetic therapy that exists now that targets the specific gene that is mutated in this patient. And so I think that by now, by our estimate, we recruited about 50% of IRD patients in Israel and genetically diagnosed many of them. I hope to be able to continue doing that. And we also apply nowadays novel diagnostic techniques that were not actually available 5 years ago or even 2 years ago, and may enable us to identify mutations that were not, that it was harder to identify before. So we're trying to use novel genetic tools in order to diagnose those patients that were harder to diagnose before, in order to achieve the highest rate of genetic analysis possible in our population.
Yeah, thank you. And there was a question posted with regards to the number of choroideremia patients, and I think from your earlier diagram, it's about 1%, Dr. Ben Yosef?
It is very rare. We have very few patients. It's a rare disease in the world, and it's a rare disease in Israel as well. I personally encountered only really a few all over the years that I've been doing this.
Yeah. And so we've had several of our researchers that have been looking at CRB1, some researchers from Leiden and Columbia and other places. Can anyone comment if you're collaborating with any of those researchers or doing anything individually on CRB1 related IRDs? Okay. But certainly it is a disease of interest for the foundation. And so we're always looking to build the research community. So as that may become something that shows up for you in your research, certainly keep us aware of that. Here's a question just really on the
Just to
the
mention about
process
CRB1.
of genetic testing. So I'm sorry, go ahead, Dr. Sharon.
Can you hear me?
Yeah.
Okay. Just to mention about CRB1. So using RNA editing, we are selecting the most common mutations worldwide that are applicable for this technique. One of which is actually a nonsense mutation in CRB1. But I have to say that this is relatively tough disease to deal with because of the early onset of the disease and the structure of the retina, which is interrupted relatively early. So this is one of the toughest genes to treat by this kind of techniques, but we're making an effort right now.
There's a very rare subtype of familial foverotinoschisis, which is a bit milder, but even that expresses very early. So it's a very tough disease to address in terms of its early and severe onset in most cases.
Amy, with your
It may
permission, I
actually
see
benefit from the other newer approaches being tested now, not necessarily in our consortium, but worldwide, including the different approaches to optogenetics, as well as the new approach by Endogena to try to awaken dormant stem cells in the ciliary body to renew the retina, or cell-based therapies. It will require perhaps somewhat different approaches.
Amy, I would like to address the Q&A question here, if that's okay. Once a patient's genetic mutation has been identified, is there any benefit for further testing, retesting? So if you were tested and a mutation or two mutations were identified, and this is a 100% certain cause for your disease as the information was given to you, then I don't see a benefit at this point to do further genetic testing on yourself. Of course, other family members can be tested, but on the patient himself, I don't see a benefit. However, if you were tested, even by whole exome sequencing, and the mutation was not identified, new techniques are being developed all the time, and now we are able to identify mutations that were not identified before. So I do suggest that if it's been a few years since you were tested and the result was negative, do approach your clinician or your geneticist and ask them to have another look, or even to perform a novel testing if possible, because yes, there is a chance that it couldn't be solved 5 years ago and it can be solved today.
Thank you, Dr. Vinyasa. We have run out of time for questions. But once again, I just want to remind the audience that if we weren't able to get to your question, or if something comes up following this webinar, you can email the foundation at info@fightingblindness.org, and we will respond to your question directly. And with that, I would like to turn the call over for closing remarks by our Chief Executive Officer, Mr. Jason Menzo.
Awesome. Well, thank you so much, Amy, and really thank you to all of you for joining us today. My name is Jason Menzo. I'm the CEO here at the Foundation Fighting Blindness, and I especially want to thank our presenters for their really interesting and fantastic presentations. We're super proud to collaborate with each of you and to continue to believe in the fantastic work that you're doing in each of your labs, and of course by your many colleagues there in Israel. I know each of our attendees enjoyed the discussion today and walked away impressed by your work, and like all of us, very optimistic for the treatments that are in development. In my role here at the Foundation, I have this really unique opportunity to see firsthand how our funding makes an impact on advancing treatments and cures for blinding retinal diseases. And I want to remind everyone that we can only accomplish what we do through the generosity of our supporters. So thank you to everyone who's supported the Foundation in the past and continues to support us every day. It's truly all of you who are our supporters who are making this happen. And I'll close with this. The Foundation is now on the cusp of being 52 years old. And in that 52 years of our existence, we have consistently been the undisputed global leader in driving the search for treatments and cures. We have raised nearly $900 million to date, and today have a full suite of programs, from basic research to a patient registry to genetic testing to natural history studies translational research, and then now through our Retinal Degeneration Fund, have a unique vehicle for venture investing in startup companies that are advancing our mission in this space. We truly are covering the entire spectrum of our mission. And again, this is only possible thanks to the generosity of our supporters. So thank you all for your support. Thank you for joining us today. I want to reiterate that if you ever have a question or if the foundation can ever be of service to you or your family, don't hesitate to reach out at info@fightingblindness.org. And our website, fightingblindness.org, has a ton of resources and information. Thank you again for joining us today. We look forward to bringing webinars and information sessions like this to you again in the future. Have a great rest of your day, and we'll talk to you soon.